Welcome back students to Yupp Master. This is your botany class and today we are going to start our new chapter, Cell as a Basic Unit of Life. Previously we have completed morphology and biological classification, plant kingdom. Now this is going to be our fourth topic. Clear? So the diversity is over, the entire of the diversity from the living world up to the plant kingdom. We have completed the diversity in the living world in terms of plants. Okay. Now today we are going to learn that how the prokaryotic, eukaryotic cells are different. That we are going to take the introductory part of this particular chapter so that we can understand that how we can classify the prokaryotic organisms separately from the eukaryotic ones. Although the cell means the thing which is present in us means the multicellular organism as well as the unicellular organism like Chlamydomonas, Euglena, eukaryotic ones, or any bacteria which is prokaryotic. Whatever be the form of organism, whichever kingdom if we are talking about, among these five which were given by Whittaker, they all have cell in them. Okay. The interesting thing about the cell is the type of the cell. But beforehand we need to know one thing, that what is cell actually, because the term cell is related to the other uses also. If you are saying watch, it is also having some cell. Okay, that is also called battery. The cell is there in chemistry and physics also. Okay, but those cells are different. The meanings are different. The uses are different. And what we used to study as cell is obviously a different one. We are studying the living cell. Okay, about the things that we say about the cell is like that if we are studying the biological organization of living, biological organization in the living world, then this biological organization is actually starting from the level of molecules. When they interacted in a medium which is called as protoplasm, and that resulted in the formation of cell. Now whatever be the kind of organism, either unicellular or multicellular, these all have cells in them. Okay. Although if anyone is asking to me that what unicellular organism means the cell which is able to survive independently is also the unicellular organism. So it is better to define cell in terms of unicellular organism. We will discuss about it, but before that, about the discovery of cell. Now the cell that we are going to study here is the cell of biology. Clear? Not about the chemistry, physics or anything else. You can say different words. What different words? See, for example, if I am saying if anything is room, if anything is mentioned as room, okay, if anything is stated as chamber, if anything is mentioned as vessel, these all can be mentioned as cell depending where you are using these words, these terminologies. Okay, let's see. First, the Robert Hooke is known for discovery of cell. He proposed the term cell. But the person Robert Hooke was studying the cork cell. He observed the cork cells. Now the cork, this cork means the same which is forming the outer body of many tree trunks, and about this cork I can say which is used in making the bottle corks. Now this cork is actually a dead material. So what this person Robert Hooke observed and mentioned in the book Micrographia is all about the chambers that he has observed. He observed these chambers. Now the chambers are empty, there is nothing inside them right now. Means what he actually observed was boundaries. He was observing the boundaries only, means he was observing something with defined boundaries. Defined boundaries in the case, the room can be defined only when it is having not only the floor and the roof. It must have the walls also, the surrounding walls. Then only you can define a room. The same is about any vessel or any chamber. Okay, means the surface, means the covering. The boundaries are going to define the shape of the vessel. Okay, the kind of the room, big room, small room, like that. Okay, big chamber, small chamber. The structure is divided into chambers because it is having boundaries. That's why. Okay, So that's why we say that the cell means a lot of things, but here cell is actually what he observed. He observed it as hollow vessels. He said hollow vessel and call it as cella. Now this is in Greek language. This is a Greek word. Cella is a Greek word. Now this cella word is signifying for what? The cell of the present day in biology. Although the living cell was discovered by Anton von Leeuwenhoek, he was the person who is noted for or who is told for the discovery of the living cell or the structure which is showing life for the first time. But this cell which we are going to define here, this structure, this concept is not associated with Robert Hooke himself. There were so many persons who were discussing about the unit that is present in our body or in the body of any organism. Means how all the organisms are same, from unicellular to multicellular, from kingdom Monera to kingdom Animalia, how all the organisms are same. They are same because they are made up of cells. So how these cells are going to perform. Now secondly, how these cells are going to be defined. How to define a cell. Now if to define a cell, firstly I need to tell about the component of the cell. Then only I can mention it. Why for this? As I said, a room can be dining room, a room can be a library, a room can be a classroom, a room can be a drawing room, a room can be a bedroom. How you are going to state these words? It all depends on what type of furniture you are going to put there. Means the furniture or the content of the room is going to define it as either dining room, drawing room or living room, classroom or a library, mess or a kitchen. Everything can be defined on the basis of the contents, the constituents, what makes the room. Okay, it's just like that so many vessels or so many boxes may be there. It's like we went to a sweet shop and we are asking them to get the sweets packed into different one. I said that just give me one box of brownie. I said one box of brownies. So the sweet box is having different contents, right? So I am saying one is having brownie in it. In the other I am going to put some jellies. In the third one, I am going to put something else like biscuits or cookies you can put. So the box may be, obviously you are saying that these boxes may be looking similar from outside, same from outside, same size, but they are different because their contents are different. So that's why we are saying the fundamental dissimilarities between the cell must be about their contents. What makes the cell? This is going to be the different one. Robert Brown was a physicist who observed the most prominent structure inside a cell and he named it nucleus. So he called that this nucleus is a well-defined structure in all cell types. Okay, so he used this criteria, or he said that the nucleus is the basis by which you can define the cell. So afterwards, different persons were there who said that if nucleus is properly defined, if nucleus is not properly defined. So there are two types of cells. How many? Two types of cells are there. These are not animal and plant cells. These are prokaryotic cells and eukaryotic cells. It is just like the nucleus means karyon. Nucleus, another meaning is, or another word for nucleus is karyon. So if nucleus means karyon, then they are saying prokaryotic means prokaryon, where the nuclear material is not surrounded by a membrane. I am just saying nuclear membrane is absent. So as the surrounding structure is absent or the limiting membrane is absent, it can't be defined properly. You can say incipient nucleus. You can just say incipient nucleus is present. Those cells are mentioned as prokaryotic cell. Whereas U means true. U means true. How true to define means it is having certain boundaries, limitation. Clear? Limiting membrane is there. Here we are saying nuclear membrane is present. So if nuclear membrane is present, it means you are able to identify this structure properly. Okay. So having the limiting membrane or having the limitations of the boundaries is helpful in creating the definition, or to say some entity as a distinct one or distinguishedly identified. That is what we used to say: prokaryon, eukaryon, prokaryotic cell, eukaryotic cell. So these are the type of cells. Two type of cells are there. Type of cell, if anyone is going to ask you, answer two. Don't say plant and animal cell. Fundamentally, these are prokaryotic and eukaryotic cell. There is another type also, mesokaryotic, but it is included in eukaryotic itself. Okay. Why? Because in mesokaryotic cell, the nuclear membrane is present. The primary criteria to categorize cells into two types is about whether nucleus is well defined or not. If this nucleus is well defined, if it is having the nuclear membrane, I can say the cell type as eukaryotic. If not, then I will call it as prokaryotic. See, the same thing we were discussing about the boxes, the sweet boxes having different contents. So the name of the sweet boxes can be mentioned: the box of brownies, the box of jellies, the box of biscuits, the box of cookies. The same is in your kitchen. Whenever you are going to move into kitchen, usually what moms used to do, they used to maintain a symmetry of boxes. They used to maintain a symmetry of vessels in which different things are kept. Rice, pulses, groundnut, these all are in identical boxes, looking identical from outside, but they have different contents in them. The same is about the room. If you are going to change the furniture, say for example, if I am going to consider this thing as my bedroom, in that cases, the first thing to be replaced is this board. Okay, this board must be replaced by something else. And if it is a library, in that cases, I need to put some shelves in which some books can be kept. That kind of classification we are going to do for the cells as well. So how the cells are classified? Cells are classified on the basis of the contents they possess. The primary thing is nucleus. Truly defined, because nuclear membrane is present, eukaryotic cell. Clear? If not, in that cases, prokaryotic cell. So a bacterial cell is prokaryotic because the nuclear material is not surrounded by nuclear membrane. Okay. So that type of cell is prokaryotic. Eukaryotic, a cell who is having a proper or a true defined nucleus, nuclear membrane around the genetic material or the nuclear material. That will be eukaryotic cell. Includes what? Chlamydomonas, chlorella, euglena, all the protistan you can take. Clear? You can come to the fungi, you can come to plantae, they all have eukaryotic cells in them. There are in total 4 kingdoms which are having eukaryotic cells. There is only one kingdom in the 5 kingdom classification of Whittaker which is having prokaryotic cell type. Fine. Prokaryotic cell type in where we can see this bacteria, and eukaryotic cell type, any of the examples in 4 kingdoms, any of the 4 kingdoms, anyone you can take, whether it is P. protista, P. plantae, F. fungi, A. animalia, any of these. Okay. So this is about the type of cells. Fine. Type of cell is done. Nucleus was discovered by Robert Brown. 3 scientists we have mentioned so far right now. One is about Robert Hooke, who proposed the term cell. Second, Anton von Leeuwenhoek, who observed the living cell. Third, Robert Brown, who reported the nucleus, the prominent structure in the cell. Okay, fine. Then how to define a cell? As I told you that it is better to define a cell in the terms of unicellular organism. Why? Because a unicellular organism is capable to perform all its activity alone. Then only I can state about cell also. Say for example, my body is consisting of n number of cells. Millions of cells are there in my body. But in that cases, I am saying that the cells in the liver are performing the function something different from the cells of the stomach. Okay? Or I can say the cells of my epithelium, means the skin, are going to perform some functions which are different from the cells of my tongue. So, right now in terms of multicellular organism, I am not getting the appropriate definition of the cell. Whereas in unicellular organism, the same cell is going to perform all the function. It is related to getting the food, digesting the food, utilizing it in the growth and reproduction and normal metabolic activities. The same cell is the place of all the type of metabolism which is required for normal survival. The same cell is going to be the reproductive unit as well. Fine. So, the cell can be better defined in terms of unicellular organism. The unicellular organism which is able to perform the activity independently, it can exist independently, and number two, that performing essential functions of life. Fine. Then I have a doubt. I said we are multicellular organism. Then why this definition of cell is not applicable to my cells? Actually, in our body, means in multicellular organism, if you want to know that what is the true definition of multicellularity, if you know the definition already, then fine. If you don't know the actual definition of multicellularity, here it is. Organisms are of two types, as I told you. Organisms are of two types. One type is unicellular and the other is multicellular. Then how to differentiate between the two? In multicellular, I have observed division of labor among the cell. I have observed division of labor among the cells. So, if there is a division of labor among the cells, it means the cells are interacting with each other. Each cell is not performing all the functions required for whole of my body. There is an interaction between the component cells in my body, and whatever they are going to perform is actually the physiology of human body, right? Or my body, right? So, in that cases, we are saying the division of labor is making multicellular organism to be the different one than the unicellular. Okay, so that's why here it is mentioned that division of labor has made multicellular organisms different. If division of labor is present, multicellularity is true. So why I chose unicellular organism to define the cell? Because those unicellular organism can perform all the functions independently, and if they are not able to perform the functions independently, all the things which they require for their growth independently, then they must get dependent on nearby cells, either as a parasite or maybe something like symbiosis, etc., that we have studied in the earlier chapter as well. So unicellular means the one who is independent, whole soul, one cell. Clear? Only one cell is going to do all the functions. Okay, so this is how the unicellular and multicellular organisms are different, and that's why I have chosen unicellular organism as the criteria to define cells, not the multicellular ones. We are better than unicellular because our cells are getting specialized to perform different functions. In unicellular organism, there is no specialization. Actually, the same cell is acting as gamete. Same cell is undergoing division, asexual. Clear? The same cell is going to perform the function of nutrition, digestion, everything. Okay. So that's why we are saying anything less than a complete cell, cannot be considered as a living, or you cannot consider it among organisms. So anything less than a complete structure of a cell does not ensure independent living. And that's why we are saying cell is the fundamental structural and functional unit of all living organism. even in my body, in multicellular also, just imagine about the condition if some person has got his liver removed because of some diseases or some ailments. If the liver is getting removed, the person is not able to perform the normal activities. Okay, they are dependent on medical facilities that should be made available for such individual. So that's why be clear about this thing, whether the organism is uni or multicellular, they must have the cells which are performing the functions. So it means all of us in this living world are made up of cells and their products. Products include their interaction as well, means interaction among the cells, like division of labor. There comes the cell theory. Those who want to state that actually the plant and animals are made up of cells and their product. The person botanist is Theodor Schwann, that M.J. Schleiden, and the zoologist Theodor Schwann, 1838-1839, proposed the cell theory. The collaboration together, their work is stated as cell theory. We are going to study some important statements of the cell theory, not all. Okay. For example, Schleiden said, on the basis of his studies on the plant cells or the plants, he said that all plants are made up of cells of different kinds and they are forming different tissues. This thing get noticed by Schwann, and he stated, although he was a zoologist, but he didn't confine his work to animal cells. He worked on plant cells as well. He told that animal cells are different because they are surrounded by a flexible covering. Nowadays, we are calling that structure as plasma membrane. And plant cells are different because they have a rigid covering around them that is called as cell wall. So he compared both the type of cells, plant cells and animal cells. It means he was studying both plants as well as animals. After which he made the conclusion that whether it is plants or animals, they are made up of cells and their units. Okay, or you can say their products. So the bodies of animals and plants are composed of cells and the product of cells. Now I have some questions about these things. Actually, the cell in biology means something which is having more or less centrally placed nucleus surrounded by cytoplasm. If it is a plant cell, it is having a rigid covering cell wall. If it's an animal cell, only a flexible covering plasma membrane is present. This type of thing is also included in cell theory. And objections arises like this. So for example, if anyone is saying unicellular organism, that's fine, but what about multicellular organism? How they originated? How you are saying that the organism like us, means the human being, is going to be formed from a single cell? Yeah, this is true. All sexually reproducing organism, including the human beings, are formed from zygote. Zygote is single celled and we are multicellular. There in the previous topic, in the previous chapter in plant kingdom, we have studied zygote may be myocyte or maybe mitocyte. If the organism is reproducing sexually, the zygote may be mitocyte or myocyte. So it means zygote is undergoing division. But these people, Schleiden and Schwann, were not able to answer such questions. My objections, or you can say that cell theory is having certain limitations or it is incomplete. It is further required certain kind of modification. So this is here we are saying that I am not at all accepting certain things from the cell theory because cell theory seems incomplete to me. My question include that how the prokaryotic cell organization can be explained, because prokaryotic cells doesn't have a well-defined nucleus in them. Secondly, the cells like mammalian RBCs, most of the mammalian RBCs, the sieve tubes in the plant cells, angiosperms, they become enucleated on maturity, means they lost nucleus on maturity. Then how come you are going to manage these things, or how the cell is able to perform the function, or whether it can be called as a cell or not? Clear? Nextly, if these things are to be answered, one more thing is how the cell originated. What came first, the organism or the cell? Means the multicellular organism was the first, or the unicellular organism was the first, or from where the life originated. So many things were there. The origin of cells. If I am multicellular, from where I got the first cell? Okay. So I need to know how the organism become multicellular from unicellular, or how unicellular organisms are becoming so much large in numbers. They don't have such answer. This question is to be answered. Obviously, these all questions are required to be answered. Then only we can consider cell theory to be full and complete. Then comes the work of Rudolph Virchow. Rudolph Virchow, or R. Virchow, modified the cell theory. He modified the cell theory on the basis of his observation. What he said, that all the cells are arriving or arising from the pre-existing cells only. Means according to him, if I say that from where the new cells are getting formed, they are saying they are getting formed from, he said that the cells are getting formed from pre-existing cells only. According to him, the origin of cell is biotic. Origin of cell is biotic, means biological origin. It's something like division. Cells are dividing. Okay, and if cells are dividing cell to cell, in that cases it is mentioned as cell lineage. So this is why we are saying that his work is much different from the earlier ones, because he was the person who is able to say the things in a true manner, that all the organisms are composed of cells and their products. Means that he is able to say the products of the cell, means when the cell is undergoing division, some of the cell may get specialized to perform certain functions. Okay, so he explained about multicellularity also. And secondly, all cells arise from pre-existing cells. He mentioned about origin of cell as well. Okay, so this is why we are saying that Rudolph Virchow is known for modification of the cell theory, which we are saying modified cell theory, or two important points of modified cell theory, not all. Cell doctrine. So this is how the things are getting changed with the time. Okay, so whenever you are asked about that how the cells are defined, it is about unicellular organism. So far now we have studied, or we have listed the name of the scientist. One was Robert Hooke, then was which one? Anton von Leeuwenhoek. Third was Robert Brown. Next Schleiden, Shawn, and now it is Rudolph Virchow. So many names of scientists. Previously, in the diversity of living world, from the living world up to the kingdom plantae, we were studying the examples. Now it is about the name of scientist. Okay, so today we have learned about 6 names, 6 different scientists. What they performed, you need to mention them separately. Okay, so here is the thing that we need to know about the content of the cell in generalized manner. Clear? It is in generalized manner. I am not at all saying that this is about the plant cell, this is about the animal cell. No, it is about cell. That can be a plant cell or an animal cell. Here is the mention how all the cells can be mentioned on the basis of their content. Say for example, if any cell is a plant cell, then the outermost covering must be cell wall. Okay, inside which will come cell membrane. Then comes cytoplasm and the nucleoplasm. Means I am going to divide the plant cell into nucleoplasm, cytoplasm, surrounded by plasma membrane, thereafter cell wall, and what? The chloroplast and plastids. Okay, that's it. If it is going to be an animal cell, then if it is an animal cell, then I will say it will have nucleus, cytoplasm, surrounded by cell membrane over, outside nothing. Clear? Then in the cytoplasm, it is having certain unique features, like it is having mitochondria. It is in plant cell also, so leave it. It is having centriole. Means animal cells are more commonly having centriole or centrosome in them. This structure is not common in plant cells. Okay, Fine, that's done. Next, if anyone is saying that what about the bacteria, how you are going to state anything or any component of the bacteria then? So here it is about the bacteria now. So about the bacteria, if I'll say the outermost covering will be named as glycocalyx, then will be cell wall, thereafter cell membrane, then cytoplasm, but not nucleus. Okay, although all of them, whether bacteria, animal cell or the plant cell, all of them will have ribosome. All of them will have cytoplasm. Animal cell is also having cytoplasm. Bacterial cell is also having cytoplasm. The same way, cell membrane is present in bacteria, cell membrane is present in animal cell, cell membrane is present in plant cell. So three things can be identified in all the type of cells. Clear? Whether plant, animal, prokaryotic, eukaryotic, whatever. Cells, whether pro or eukaryotic, plant or animal like, same in, or you can say similar, because of occurrence of one, the plasma or cell membrane. Number two, cytoplasm. And number three, ribosome. Very important. Okay. So the cell types that we are going to study in the coming classes will be prokaryotic and eukaryotic, and obviously we are going to learn about how the plasma membrane, cytoplasm and ribosomes are similar in all. So this is how the content is going to be useful. That what content is present in plant cell, animal cell, what content is present in prokaryotic or eukaryotic cell. This can be used in categorizing or making the cells into different categories. Okay, fine. We are going to have this table. Now NCERT book has given the three examples. One, human cheek cell. You can get them very easily. Take a slide, rub it inside your cheek, you will get certain cells on it. Dead, alive, both. The second type of cell they are giving is onion peel. Clear? The scales of onion. And third type is a bacteria. Okay. I have to mention these three things or these three types. What three types? Human cheek cell, onion peel cell, and one more, and that is what? Bacteria like Escherichia coli. I need to fit them into A, B, C. Okay, let's one after another. Say for example, as I told you that plasma membrane is present in all cell types. Clear? As I just told you that plasma membrane is present in all cell types, then it means it is in A, it is in B, it is in C. All three will have the plasma membrane. Okay. Just now I told you that ribosome is also present in all cell types. Okay. Ribosome, ribosome, ribosome. Fine. But the cell wall is present in plant and in bacteria. Cytoplasm is present in all. Okay. And nuclear membrane is present in plant. Clear? Nuclear membrane is going to be present in plant cell and animal cell. Plant cell, animal cell. But animal cell is having centriole in them more commonly. That is not present in plant cells usually. So next thing, centriole absent, nuclear membrane absent, membrane bound cell organelles absent. Now you can categorize whether it is, which is A, E. coli, onion peel or human cheek cell. Coming to the next one, the membrane bound cell organelles are present but the centriole is absent and cell wall is absent here. Membrane bound cell organelles present. So if now categorization is to be done, then I can say very clearly that this cell type must be prokaryotic. Why? Because nuclear membrane is absent, then the cell type is prokaryotic. And nuclear membrane is present, it means the type B and C are eukaryotic. To which kingdom the onion plants belongs to? Plant A. They are eukaryotic, no doubt. So I need to put the name as A, B, C for these three. So definitely the C must be human cheek cell. Okay. Second, this B must be onion peel cell. And the A is Escherichia coli for sure. So remember this one, that how the things are to be defined. In a brief note, plasma membrane means a flexible covering. Cell wall means comparatively rigid. The cytoplasm means main arena of cellular activities, means whatever happened in the cell is actually happening in cytoplasm. The molecules that are responsible for functions of the organism, they all are interacting in the cytoplasm. So this is how we can mention the characters. Okay, so that's why we are saying there are three different categories we made, although you can brief them into two, prokaryotic and eukaryotic one. This is how we can categorize the cell on the basis of their content. So from where we started in the beginning, we are concluding the same thing here. Okay, we have classified the cells on the basis of their constituents. Okay, this is the way, right? So we have two different type of cell to be studied, prokaryotic and eukaryotic first. Now what are they and how these are different or similar, that will come in the later classes. First is about the prokaryotic cell. Now the prokaryotic cell means, the prokaryotic cell is something which we are saying the prokaryotic cells are represented by bacteria like blue-green algae, mycoplasma, PPLO like organisms, generally smaller and multiply more rapidly than the eukaryotic cells. They are varying in their shape. They may be like what? They are like rod shape. These are rod shapes which are mentioned as bacillus. These may be spherical, which we used to write coccus, plural is cocci. They may be like a coma, which we write as vibrio. They can be spiral or twisted, which we used to write as spirillum. So these are the four general shapes, out of which we know this thing. Most common is bacillus. This is most common type. Coccus are second most common type of bacterial cells. Okay, so this is what we know, what we need to tell about the prokaryotic cell once again I said. Okay, fine. So next is about the general characters of a prokaryotic cell. Now what are the structures that are present in a prokaryotic cell? Just recently I mentioned all the components in the cell, and now I am going to label it in the structure that is given here. Okay, so one after another one, from the outermost to the inner side. The outermost is glycocalyx. This black covering is glycocalyx here. The function we will study later. then comes the cell wall. Okay. Thereafter is plasma membrane. The plasma membrane may be having certain infoldings. These are called as mesosomes. They may have the cell organelles which are called ribosomes, which are alone or so many units of ribosomes are attached to one mRNA during protein synthesis. So these are so many ribosomes that are present on the mRNA and performing protein synthesis. So here we will call them as polyribosome or only polysome. This is a structure which is not having any boundaries or not separated from the remaining cytoplasm. This is the matrix of the cell that is called as cytoplasm, the fluid. Okay. And we are saying that here so many other things are also there which are not surrounded by any membrane. One is the genetic material of bacteria, this one, which is named as nucleoid. Obviously DNA, double-stranded DNA, the single molecule of DNA forms the genetic material of bacterial cell. It is getting coiled, it is circular one and is named as nucleoid. Whereas some other DNA particles are scattered. Several other DNA molecules are present in the cytoplasm of a bacterial cell, but smaller to nucleoid. They are called as plasmid. Some inclusion bodies, inclusion means included in the cytoplasm because they are not getting separated by a limiting membrane. So these inclusion bodies may be something as gas vacuoles or the structure which is providing buoyancy to a floating cell. Gas vacuole. So this is gas vacuole, this is nucleoid, this is ribosome, this one is polyribosome, the matrix is cytoplasm, this is plasmid. So these things are present in a prokaryotic cell. The structure is so much simple. Here we are saying the structure is so much simple that we don't have many things around them or many things in them. Membrane bound cell organelles are absent. I repeat, membrane bound cell organelles are absent. Whatever metabolic activity happens, it is happening mostly in the cytoplasm without any defined space. So many of the time we say the prokaryotic cell as single envelope system. Just now what I said, cytoplasm as main arena of cellular activities. So that is surrounded by only and only one kind of envelope. That's it. That envelope is consisting of glycocalyx, cell wall and plasma membrane. Three things. These are considered as three layers of the cell envelope. It doesn't mean the three layers of envelopes. I am saying these are forming the envelope with three layers, not the three layered and not the three envelopes. Remember, three layers of the same envelope. So the single envelope system, prokaryotic cell. Now this prokaryotic cell can be stated with the prokaryotic cell. All of them have a cell wall. Certain exceptions are there like mycoplasma. Mycoplasma is mentioned as CWD. At the time of biological classification, I told you CWD stands for cell wall deficient. Else you can say that all prokaryotes have cell wall. Okay, if you are reading the previous or the older NCERT, it is saying all prokaryotes have cell wall. Now in the recent edition it is added, and it is now read as all prokaryotic cells have cell wall except mycoplasma. Exception is there. Okay. The second thing, the fluid matrix is called as cytoplasm, and the cytoplasm is not having any membrane bound cell organelles. Clear? Without any membrane bound cell organelles. So if anyone is saying that cell organelles are present in prokaryotic cell, answer is yes, yes, present. But membrane bound cell organelles are absent in bacteria. Clear? Cell organelle present. Which cell organelle is present? Ribosome. Okay. Fortunately, ribosome is not having any limiting membrane around it. Next, the next criteria or the next feature is that the genetic material, there is no well defined nucleus. So here we say the genetic material is basically naked, means no covering. That's why. Clear? Because nuclear membrane is absent, we are saying the genetic material is naked. The nucleoid is naked, not enveloped by a nuclear membrane. Okay. Beside this DNA which is forming the nuclear material or the genetic material of the bacteria, there are, means in certain bacteria we are observing, in many such examples, plasmids are present. Now these plasmids are different. You can't say the plasmids exactly as the nucleoid. Plasmid and nucleoid are two different things. They have different significance as well. Okay. So this is about the general description. Beside this, we have this body that is called as cell inclusion. The cell organelle, that is ribosome. That's it. Sometimes the bacterial cell may be having flagella on the surface for locomotion, or maybe having other structure like pili or fimbriae type of things. We are going to add that particular things to the next diagram, not here. Okay, they are a bit different from this. Okay, we need to study the other structures as well. Consider that those structures are not mentioned here. We haven't mentioned all the structures possible in a bacterial cell. We are studying some typical structure first of all, general. Then will come the specific one. So here is the thing that I need to differentiate among it, plasmids and nucleoids. Are the two things same? Answer is no. Let's see how. So I have two words. One is nucleoid and the other one is plasmid. So nucleoid and plasmids are not exactly the same thing. If they are not exactly the same thing, then chemically both of them are DNA. Nucleoid means first of all double-stranded circular DNA. Okay. This single DNA means how many in number? 1 forms the genetic material okay, this is going to form the genetic material, or in other ways you can write it as genome of bacteria naked in cytoplasm. Fine. Whereas plasmid means double-stranded circular DNA, no difference, same, but differ in terms of what? Several molecules, or you can say several copies is a better word, several copies but smaller than nucleoid. Okay, and we say they are not the genetic material. So they say that this is not important for normal growth and metabolism of the bacteria. Secondly, that's why they are saying if it is waste, then why it is present. Sometimes they are becoming useful in the function. So they are semi-dispensable. Dispensable means waste. Semi-dispensable means can be used, can be utilized also, but not always. Q. Semi-dispensable or become significant in some special functions. By this way I can state that if nucleoid is naked DNA, then plasmid means scattered DNA. For the single means the singular. For the singular you cannot say scattered. If they are plural means many in the number, they can be mentioned as scattered. Okay. None of them is going to have a limiting membrane around them. But plasmid is having some typical role that you will study in the later chapters, that is called as genetic engineering or biotechnology. There we used to study the significance of plasmids. They are more significant in genetic engineering actually that we are doing right now, or the modern biology is exploiting these plasmids. Okay, these are of natural occurrence obviously. So this is how the nucleoid and the plasmid, the two DNAs in the bacterial cell are different. Okay, so this is about the general description of the bacterial cell, or we can say the character of the bacterial cell. So next to study is about the differences or at large how the bacteria are getting classified into two groups, or the prokaryotic cells can be divided into gram positive and gram negative, and what other structures do they possess, so that we can compare the prokaryotic cell in much better way with eukaryotic one. Okay, that is going to be our next topic to be covered in the coming class. For this class, you are obviously advised to attempt the daily practice paper and go through the questions given in the paper and assess yourself that how much you are retaining from the class itself. Yupp Master is providing that particular strategy to get maximum score, to get maximum mark with the proper planning and its execution. You can be more efficient and accurate by doing practice, just followed by class also. Okay, so there is it, means read the NCERT, follow the class, make the class notes, do the daily practice paper and attempt the weekend exam also. By this way you can retain most of the things whatever you have studied in the class. This is what we need to score the best rank in NEET 2022, and those who are seeing us and are aspirant of NEET 2021, they can also follow us on the Yupp Master, they can follow us on the YouTube live, so that they can achieve the better scores than the NEET 2020. Hope you are liking our classes. So be continuous with us in your studies and be safe at your home. All the best. Thank you.